Okay, I want to talk a little bit about the first half of the reproductive systems chapter. In this one, I'm going to do in a little bit of different order than the book does. I'm going to talk about how sperm develop, this process called spermatogenesis, talk about how male sexual response and development is hormonally controlled, and then also a little bit about sexual response and ejaculation, which is, well, sexual response is sort of the genteel way to talk about erections. Anyway, so to talk about spermatogenesis first, remember that we have learned different kinds of cell division. There are really only two kinds. The majority of cell division that happens in your body, like for growth or for replacement of worn out cells, is this process called mitosis, which has one round of cell division in which you end up with basically identical daughter cells from the original parent cell. Now, on the other hand, meiosis we know is different because it's different in a few ways. First of all, there are two rounds of cell division where, the cells divide twice. However, they don't double up their DNA between those two divisions. So, two rounds of cell division. At the end, all of the daughter cells in meiosis are genetically non-identical, right? So the cells are different genetically from one another through this process called crossing over. And the third thing that's different is those four daughter cells are all haploid. So they all have only one copy of each of our chromosomes rather than two copies, which would be typical of mitosis. I'm not going to get into the reads too much about meiosis. I have a feeling most of this is pretty familiar from being around. Really, the thing that's different is the crossing over that happens during meiosis 2, which happens where sections of DNA of homologous chromosomes actually come across one another and end up kind of breaking off and reattaching to the opposite chromosome, which is going to give this genetic difference among all those cells. Now, spermatogenesis, when we look at it overall, has two phases. There's an early phase that's the meiotic stages, and that has to do with like the genetic recombination, basically all the genetic changes that happen to the nucleus. All those things are happening during that early meiotic phase. Then later, there's this process called spermiogenesis, where the cells are no longer these, roughly cuboidal or roundish cells, but actually become a little more streamlined, lose excess cytoplasm, grow their tail, all those kinds of things. So that's spermiogenesis that comes later. Now, all of these processes happen inside the testis, but particularly they happen inside the seminiferous tubule. So when you look at this figure in this micrograph, you can see there are a few cells close to the lumen that have this sort of characteristic like arrowhead or bullet-shaped kind of structure to them. But the ones around the periphery of the seminiferous tubule, they tend to be a little bit more rounded because you can see chromosomes present rather than just like the loose chromatin of these other cells. You can tell that that's where the active cell division is taking place. Now, not all of the cells that you see in the seminiferous tubule are actually going to turn into sperm. There are other cells that are called sustentacular cells that give support to the cells that are developing into sperm. And that's not the only thing that's in the testis either. In addition to the seminiferous tubules, there are also these clumps of cells in between them called interstitial cells. If you recall back to the follicular cells of the thyroid gland, remember there are parafollicular cells as well that are making a different hormone. These are similar. In this case, the follicular cells, their responsibility is for producing sperm. But the interstitial cells, their job is about producing hormones, particularly androgens like testosterone. This is actually kind of a cool electron micrograph of what the seminiferous tubule looks like. But again, you can see those a little bit more clearly than in the regular stain, the probably H&E stain here. You really can't see any sperm tails here, but the tails are like much more clear in this case. And like we talked about in lab, the sperm start off being they become more mature as they go toward the lumen, and then once they're done, they basically, sometimes they can swim away, but more often than not, there are just this sort of a movement of fluid by way of cilia that's going to just kind of brush the sperm eventually to get out to where the epididymis is. Now, if we look at a cartoon version of the wall of the seminiferous tubule, there are a few characteristic things to point out. First of all, notice that there's a difference between what's called the basal compartment around the periphery of the pipe and then the adluminal compartment that's closer to the lumen, as you'd expect by the name. Now, all of these purple cells here, those are spermatocytes. Those are cells that are going to be turning into sperm. They start off being sort of connected to one another, but then as they grow and develop, they're eventually going to get separated. What looks like kind of a big yellow blob, like here or over here, these cells are sustentacular cells. Those sustentacular cells are the ones that are supporting them. So they're large cells compared to the spermatocytes. And again, you can see some of their nuclei in here. So really, we're only looking at a couple of sustentacular cells. And notice that there are like tight junctions between them too to allow kind of a permeability barrier. The way that this works is in the basal compartment, there are basically what we call type A and type B daughter cells. When each of those stem cells divides, when the spermatogonium is called, that's a stem cell, divides, one of those daughter cells, the A cell, will stay in the basal compartment, and then the type B cell is the one that's going to undergo other, further meiotic changes in order to become sperm. Another thing to note about this is part of the reason why there are tight junctions between the sustentacular cells is, remember, the sperm that are down here in the adluminal compartment are genetically non-identical to the rest of the cells in the body. So actually there is what's called the blood-testis barrier in order to prevent the immune surveillance of the sperm. Because basically the body's immune system would recognize them as foreign and try to act against them. So this is part of the way that that's avoided. Looking at spermiogenesis, it takes place over a few weeks, and where these cells basically start off as relatively round, and then as they develop, they start to grow this characteristic flagellum, and then what's shown here, excess cytoplasm, is basically going to get subsumed into fuel and materials for being able to operate the sperm flagellum. And if you look at a mature sperm, what we see is, so there's this nucleus here, in the midpiece, which is loaded up with mitochondria, and then the tail, which is going to be for propulsion, obviously. And at the very end of the head, there's a structure called the acrosome. The acrosome is filled with digestive enzymes that will help the sperm to be able to fertilize the egg when it comes across it. There is pretty steady hormonal control of spermatogenesis in guys, and this is regulated by way of the Hypophyseal Pituitary-Gonad axis. In this case, releasing hormones, GnRH, will come down to the anterior pituitary and cause those somatotropes to release FSH and LH. Now, these two hormones have different targets. Follicle-stimulating hormone goes to the sustentacular cells and is going to bind first with a hormone called ABP, which is called androgen binding peptide. And ABP is going to kind of upregulate the activity of the sustentacular cells, as well as kind of increase the meiotic rate of the spermatogonia. Luteinizing hormone, on the other hand, is going to go to the interstitial cells, and they're going to make testosterone. Testosterone is part of the feedback system too. So when LH causes the interstitial cells to make testosterone, also affects the rate of sperm production. But testosterone feeds back on the anterior pituitary and shuts off the production of LH. Similarly, when ABP is bound, it's going to release another hormone from the sustentacular cells called inhibin, which is inhibitory for GnRH as well. So that's also going to kind of shut off the FSH realm. So this is kind of the regulatory system The reason why it's called an axis here is because basically the hypothalamus communicates with the pituitary, which communicates with the testis, and then the testis feeds back to say, okay, I got the message, either by sending out testosterone, the product of luteinizing hormones, target cells, or by releasing inhibin, which is the the response that comes from the sustentacular cells. Yes, this is maybe my favorite graph in the book. What we're seeing here is the production of testosterone. So in red, that's the plasma levels of testosterone shown like before birth and then up to, well, your eventual decrepitude at the end. And then sperm production, which is shown in that kind of dashed blue line. So sperm production doesn't start until puberty, but notice there's some pretty high spikes of testosterone early in development, which is primarily responsible for like when the initial production of the testes and the penis and the scrotum and stuff like that is initially happening. There's another kind of early burst around just after the time of birth, but then it's pretty flat through most of childhood, and then ramps up. And that red line, the sort of, it's not exactly dimensionless. Notice this is like a kind of a percent scale on the side. So when you get to adulthood, in your like late teens, at least your hormonal adulthood in your late teens, that's going to have a pretty high rate of testosterone production and sperm production for the great majority of your life. Notice there's like this 40-year break in scale here. Then it's going to kind of start to drop off after when you get into your mid-60s or so. But yeah, it's pretty much a wash of about 400 million sperm per day that are going to get produced. Now, where these sperm go is ultimately going to start to hang out in the epididymis, which is sort of this holding pen sports bar where the sperm will sit until the time of ejaculation. What happens is the sperm, when they're released, will kind of move their way one way or the other down these kind of this cul-de-sac of the seminiferous tubule, end up in this little network called the rete testis, and then from there get kind of moved along by cilia into the epididymis, where they're going to sit here and kind of get stored. So they enter down here and they'll kind of be stored in this like comma-shaped loop here, but it isn't until they're going to be sent out for ejaculation that they that they're actually going to get, but that they're not swimming out during ejaculation. They're getting sent out by the action of muscles, smooth muscles. They're going to be sending them along. This kind of busy figure is showing the route that the sperm would take on their way out. Now, so first the sperm, which are in here, this holding pen of the epididymis, are going to, in the initial phase of ejaculation, they're going to get sent out through the ductus deferens and then into this little kind of bulb-like ampulla, and they're going to join up with this very short distance, a structure called the ejaculatory duct, which then empties into the prostatic urethra. And then the sperm basically make it all the way to the end of the urethra in that initial phase. Now, the main like propulsive part of ejaculation happens when both sperm are present, as well you get the excretions of the seminal vesicles, as well as the prostate and the bulbourethral glands, or what used to be called Cowper's glands, these little bitty glands in the urogenital diaphragm, that are all releasing different constituents of the semen. So in terms of volume, there's a very small amount of volume that's coming out of the of semen that's actually sperm. Most of what's there is coming from the bulbourethral glands, the prostate, or the seminal vesicles. I'm going to refer you back to the textbook rather than have you take notes off this for what's actually getting produced by each of those by each of the accessory glands. But the thing to understand is, in general, this is the semen altogether is to try to help the sperm to activate themselves and to live as long as possible. So there's like food that's sent off. It's a pretty alkaline semen overall to try to counteract the more acidic vagina and uterus. And also there are some slightly acidic substances that will that are responsible for kind of activating the sperm. But as far as actually one point, I think this is sort of a close-up of this section of this figure. Just to remind everybody that the two paired ductus deferentia are coming together with the ducts off the seminal vesicles, into the prostate, and this little intersection here, where all four of those pipes are coming together, that's where the ejaculatory duct is. The ejaculatory duct sounds like it should be like the big name for all of those pipes, but it's not. So that very short distance goes into the prostate. Oh, the old literature will call the prostate gland, though the prostate has glandular functions. It makes this, a lot of the stuff that's this kind of milky alkaline fluid that's coming out in the semen. It's also got smooth muscle in its wall, so it's actually what's responsible for most of the contractile force of ejaculation, for propelling the sperm out. Now, how erection happens is there are these, you remember that there are these bodies of erectile tissue here in the penis. There are the two paired corpora cavernosa with these deep veins, deep arteries in the middle, and then this the corpus spongiosum, which kind of goes around the urethra on the ventral side of the penis, and also will erect the glands of the penis as well. And those things have actually slightly separate. The two paired corpora tend to get themselves erected together, but it can be a little bit different from what's happening with the corpus spongiosum. What happens is this in order to allow intromission, in order to allow coitus to be able to happen, the penis has to be erect, otherwise it's not going anywhere into the vagina. The way that the erection actually happens is there's a dilation of the deep arteries of the corpora cavernosa as well as the corpus spongiosum, and at the same time, actually largely through the work of nitric oxide, which is a vasodilator. And there are these, I don't have a good picture of it. I'm going to have you watch a video later of what the morphology of the corpora is like, but it's almost like a helical kind of cross-linked collagen and elastin container that the erectile tissues are surrounded by, so that when they enlarge, they don't gonna go circular. They end up becoming more cylindrical in their shape. The result of this is that the corpora will slowly fill up with blood, and there's this tension on the back end where there's basically prevention of the blood from leaving. So as a result, the incompressible liquid of the blood is going to be what's responsible for causing the erection to happen. Just before ejaculation, also there's involuntarily there's a kind of a clamping down of the internal urethral sphincter. So so basically there won't be any mixing of urine with the with the ejaculate. In fact, part of that early phase of ejaculation, one of the things that happens is the Cowper's gland releases this substance that kind of neutralizes any residual urine that's already in the urethra. The way the two crura of the penis are attached to the pelvis, they're attached to the ischium. Actually, I suppose I should go back. The way that these two crura are attached to the ischium and the bulb is sort of attached to the pubis, that's what's going to keep the penis from getting shoved into the body during intercourse. Looking in this sort of 3D picture, sort of like the figure from the models from our lab. So again, the sperm are coming up, kind of hooking around the back, sort of posterior to the trigone. They're going to merge up through this ejaculatory duct and then merge into the prostatic urethra and then head out. One thing to remember about the process of ejaculation is there are these three phases. There's that first phase where the sperm are released and basically make it all the way down the pipe, and then the main propulsive phase that's mainly done by the prostate, like I said before. And then last is this sort of kind of squeegeeing out phase that happens where any kind of residual sperm is going to get kind of mopped up as the erection dies back. Anyway, that's almost everything I want to say about that. Just a little note about morphology. We're going to talk a little later in the next video about how the testes descend. But recall a little bit about the morphology of the scrotum here. So all the stuff we've been talking about has been pretty deep. However, there's this one sling that kind of is derived from peritoneum as well, that's going to surround both the cremaster muscle that goes around each testis, as well as this layer of this kind of tight layer that's called the tunica vaginalis, that's going to go around the individual testis, and then all of the testicular artery, the pampiniform plexus of veins that turns into the testicular vein, as well as nerves that go down to the testis. All of those are going to kind of get slung within this spermatic cord. There is a septum in the middle of the scrotum, you probably recall, so like the left one never like becomes the right one. And also there's another muscle along the kind of outside of the scrotum called the dartus muscle, in order to kind of which assists in kind of hugging the scrotum closer to the body in case it's too cold. Though the testes have these two functions, the functions producing testosterone and also making sperm, as it turns out, normal sperm development happens at a slightly lower body temperature than the core temperature. So that's a way for the body to be able to regulate the temperature of the temperature of the testes, so they're a little bit cooler than core temperature. In addition to that, there's actually also some work that's been done that's not only the temperature, but also the pressure inside the abdomen that can be a little bit too much for sperm to develop normally. I don't have a good resource to give you on this, but stuff that's happening with work in primates is we're kind of understanding now that it's not just the temperature, but also when you think of the weight of the organs that are kind of, packed into the abdomen and pelvis, the sperm are pretty, can get killed off pretty easily by like tons of extra pressure. So that seems to be like another evolutionary explanation for the development of the scrotum, because like not all primates have them. Not all mammals have scrota either. So it seems to be related both to the temperature as well as the pressure as a result of locomotion. Anyway, that's the main stuff I want to talk about with this. Next up is going to be the female side.